This model shows transcription for eukaryotic type 2 genes which encode mRNAs that make proteins. In this example, we have the gamer gene, which encodes mRNAs that make proteins that make gamers exceptional Pac-Man players. The blue strand is the template strand, which in our diagram runs from 3' on the left to 5' on the right. The coding strand is represented by the black dots and is anti-parallel to the template strand, meaning it runs from 5' on the left to 3' on the right. Here we have labeled various cis-acting elements. We have BRE, which is B recognition element, on either side of the ta-ta box, which are denoted by BRE-U and BRE-D for upstream and downstream. BRE helps stabilize DNA after binding with the transcription factor TBP. The ta-ta box is the consensus sequence or the primary element that is recognized. Next, we have the initiator region, or INR, which includes the transcription start site and is located from -2 to +4. Further downstream, we have the motif 10 element, or MTE, which is found in some mammalian promoters. That is located between +18 and +17. And even further downstream, we have the DPE, or downstream promoter element.
To begin transcription, we will first form a pre-initiation complex, or PIC. The pre-initiation complex will be formed using various proteins called transcription factors. Our first transcription factor that binds to the DNA is called TF2D, or transcription factor 2D. It associates with the DNA with two important components, TBP, or ta-ta binding protein, and TAFs, which interact with the INR and DPE region. Then TF2A stabilizes the complex along with TF2B. TF2B specifically binds to the BRE-U and the BRE-D regions of the DNA. Next, TF2F and RNA polymerase associate away from the DNA. TF2F brings in RNA polymerase to the rest of the complex. Finally, TF2E and TF2H bind to the pre-initiation complex. Both of these transcription factors are important for melting the DNA into an open complex form.
Now that we have our open PIC, let's talk about the regulation of transcription. So first we have the mediator complex. This is the adapter molecule that causes looping of DNA, bringing general transcription factors closer to the regulatory factors. It directly interacts with RNA polymerase II, TF2D, TF2H, and TF2E. Apart from the mediator complex, there are other cis-acting and transacting elements that contribute to the regulation of transcription. First, we have the proximal regulatory elements that spans from -200 to -50 of the DNA. This region has two consensus sequences, which is the GC box and the CCAAT box. Any mutations in these two consensus sequences can reduce the rate of transcription. Apart from this, we have various response elements which are also present on the DNA. These enable genes to respond to specific factors. For example, we have the GRE, or the glucocorticoid response element, which is the recognition site for the hormone glucocorticoid receptor. Next, we have the distal regulatory regions, which can be present thousands of base pairs upstream or downstream of the transcription start site. These regions include enhancers, which increase the rate of transcription, and silencers, which negatively affect the rate of transcription by binding repressors. Next, we have the insulators, which are nucleoprotein complexes partitioning different genes. Next, we have the LCRs, which are composed of all of these cis-acting elements which collectively regulate the entire locus. So all of these regulatory factors have to act together for a successful initiation event. So once we have a functional open PIC, the RNA polymerase begins transcription. Once it successfully adds 25 nucleotides to the nascent mRNA, the capping enzyme adds a 5' cap to the upcoming mRNA, which protects the 5' end of the mRNA from exonuclease activity.
Transition from initiation to elongation involves the phosphorylation of the carboxy terminal domain at the serine residue. RNA polymerase proceeds to read the DNA template, adding nucleotides and extending the RNA strand in the 3' direction. There are transcription factors that regulate elongation, such as positive transcription elongation factor B, which promotes transcription, and negative elongation factor DSIF and GDAL1, which blocks elongation. Once the cis-acting poly-A signals, AAUAA sequence are transcribed into the mRNA. The CPSF and the CSTF transfer from CTD of RNA polymerase II to the poly-A signal. These two factors then recruit endonucleases to cleave 10 to 20 nucleotides down from the poly-A site. Poly-A polymerase then catalyzes addition of approximately 200 A residues at the 3' end. This is known as polyadenylation. RNA polymerase II continues to transcribe for several kilobases, eventually dissociated from DNA.